Hello, Smita. How are you? Hello. Hello, Smita.
Hello, ma'am.
How are you?
I'm fine. Thank you so much.
Good afternoon.
My school is starting IA in biology, and I do not know what to do, how to go ahead. So, can you
Okay.
please guide me with respect to the biology HLIA?
Yes, definitely I can do it. You have joined this year. You are in DP1 right now.
Uh, yes, I'm in DP1 right now.
Okay, okay, fine. Now, what we are going to do is that we will I'll share the screen first with you, and
Okay.
you can ask me any questions, whatever I'm explaining. If you don't understand, you can
Yeah.
ask me, and
Okay.
you can raise your hand and ask me, and I will answer your queries accordingly. Okay, let me start sharing the screen with you. Can you see my screen, please?
Yes, yes.
Okay.
Can you make it little big?
Uh, this is this is the maximum
Yeah, yeah,
size.
yeah. I can see. I can see. Thank you.
Okay, okay, fine, fine. Now you can see.
Yes.
Okay, fine. Let me start saying that the biology internal assessment. Now, internal assessment is a part of the DP program. Uh, there are so the assessments in DP are two types: internal assessment and external assessment. So, what is it? So, internal assessment constitutes some weightage, and external are the papers which hold some weightage. So let us talk about the biology internal assessment. As you have taken biology in the DP, so biology internal assessment is called as the scientific investigation. Okay. Now, this is the name given to the internal assessment, and the duration you have to spend, the duration is 10 hours. Now, this 10 hours is the practical work or the secondary data you are taking, you're doing the analysis, that is the 10 hours. And the maximum marks is 24, and the weightage is 20%. So the 80% is external assessment, that is the papers that come from IB. Okay. Now, how are you going to? I'll give a rough idea starting and then I'll go into the detail of it. Now, this, whatever the scientific investigation that you're going to do in the 2 years program, that you have to submit to the IB in the form of a written report. Okay, a report has to be given to the IB, and this report has to be 3,000 words. You have to be very, very particular in this regard. It should be 3,000 words, and you cannot go beyond that, otherwise, you will be penalized. Now, the whatever your report you send, it will be first internally assessed by the teachers, and it is externally moderated by the IB. That means the teachers will mark you in the school according to the criteria, and then it will be sent to see the moderation has been done to see whether the marking is done correctly, accordingly, or not. Now, what does the IA, the written, that is the scientific investigation written report, are the things that are not included in the word count. Okay, they are the charts, the diagrams, the charts, the tables that you make, the equations that you write, the formulas, the calculations, the bibliography, the citations, referencing. It can be footnote, it can be in text citation, anything, bibliography, headers. These are the few things that are not included in the 3,000 words. It is the report that you have to write, and there is no need to have a cover page or the content page. This you have to be very clear, because I have seen many students putting the cover page and the content page, which is not at all a requirement, but at the and also these are excluded.
Mm-hmm.
Now, next, now let us start slowly in, we are getting into the IA. Okay, now the most of the children or most of the students find it difficult to formulate the research question. They
Yeah,
are
yeah. That was
they
what I wanted to ask you. Yeah.
yeah, yes, so they don't know how to do a research question or what needs to be done. So what happens, these are the few tips I have written over here, which are that the topic that you choose in the biology, because you're doing the biology internal assessment, so the topic that you choose should be of your interest. You should like it. Okay, any topic you think. It can be from the field of microbiology, it can be in the field of what is called as enzymes, anything that you do that has to be of your interest. Now, when you choose that of your interest, the IA should be unique. Unique in the form of topic. The topic that you choose should be a unique, or the method, that is a procedure that you're going to follow for bringing the data and all those. Either this should be unique or the analysis has to be unique. This is something that the moderators are looking into it. And the third thing is that the topic that you have chosen should be applicable to the either to the global context or to the local context. That you should be very, very clear about it. And they should extend the prior studies. That means whatever your prior knowledge is, that it should be an extension of that. The fourth point is you must be able to justify why you're choosing. So when you're writing the background information at the end, you should, when you're before putting the research question in the background information, you should write, you should justify why you're choosing this topic and why is it important, why is it relevant. That you have to do. And the last one is you cannot repeat the IA ideas from your other courses. That means, like if you do vitamin C estimation, okay, in chemistry also there is vitamin C. So you cannot do that. Nor can you have an IA similar to your peer in the same class. Okay, your IA should be unique. So these are the tips that for coming to formulate a research question. Now, how I am going to do is that now there are four criteria, okay, for the IA. Okay, that is, one is research design, which I put it over. The second one is the data analysis, which I will show you. The third one is conclusion, and the fourth one is evaluation. So these are the four criteria, and each criterion is of 6 marks. So 6 into 4 is 24 marks.
I'm
So
not able to see the criteria here. We are just stuck up on the second slide only.
okay, can you see it now?
No, second, you have to click maybe the third, fourth slides. On which slide are you teaching right now?
Yes.
Which slide are you teaching right now?
Third. The I
Okay.
have now I have come to making sense of the criteria.
So
Can you
no,
see it?
this is still showing the second slide.
Okay, let me see where. Okay,
Now I can see third slide. Click on the third.
okay.
Yeah,
Now,
thank
can
you.
you see
Yeah.
this one?
Yes, thank you.
Yeah. So now this is the research design. This is the first criterion, A criterion. There are four criterion. The B criterion is data analysis. The C criterion is conclusion, and the fourth criterion is evaluation, which you will see in due course of time. So the
Are
first
you are you are
criterion,
you on the fifth slide now?
yes, ma'am.
Okay.
So so this criterion has got three aspects. One is the one which I've written in the white. That is, the research question is described within a specific and appropriate context. This is the second criterion. Now, the aspect. I will explain to you each aspect, and then I will show you a sample also, so you will understand in a better way. Now, this is the first aspect, which says that it should be described within a specific and appropriate context. Now, you know what is context. When you are taken a topic, you have to have an independent variable and the dependent variable, and the background information should be appropriate or should be relevant to the topic that you have taken. It should be in context. Why? The reason is that the background information will be used when you are doing the what is this one, the background information, you will be using it when you are doing the analysis part. Okay, so this part, this aspect, this first aspect, you have to be having the dependent and the independent variable. Besides that, the background information should be there. Now
Can
coming
I
to
ask
the
a question here, ma'am?
yes, please.
How many independent variables and dependent variables should
Yes.
biology IA have?
Yes, the very good question, Smita. Only one independent variable and one dependent variable. The reason is that if you take many, you don't know which is relevant, what is affecting it. I will give you an example. For example, if it is the rate of photosynthesis, we are doing the experiment on rate of photosynthesis. Now, there are many independent variables. You can take temperature, you can take carbon dioxide, you can take water, you can change the quantity. Okay, you can have different temperatures, you can have different amount of carbon dioxide and different volume of water. But what happens, my dependent is the formation of oxygen. Okay, now if I take these three independent variables, I will not know which independent variable is affecting the oxygen production. So I have to have one independent and one dependent so that I know that this is the factor which affects the oxygen production, and to what extent is it affecting? Is it increasing or is it decreasing? So it is always preferred to have one independent and one dependent variable, or if you are taking two independent variables, then you should have two dependent variable also.
Okay, thank you.
Okay. Now coming to methodological. Now, as the name suggests, this means that it is a procedure. Okay. So research design. Okay. Design, as this name suggests, first of all, you have to have the research question. Now you have to put the method in place. So when you're writing the method, you have to write the procedure, the materials required, everything has to be done over here. And then this is very important, which the children miss it out, the safety, ethical, or environmental issues. Okay. Now, ethical, if you are using a plant or if you are using a what is called as an animal, then how are you making the animal undergo a strained condition? This is very, very important. And safety, like how you're disposing of the chemicals, are you diluting it, how you're using it, all these things should be considered in this second aspect of methodology. Now coming to the next slide, is it visible?
It's not gone to the next slide. It is now in number 5.
Okay.
You have to click on number 6 now.
Okay.
Yeah, thank you.
Yeah. So now this is the third aspect of the research criterion, which says that which says description of the methodology. What do you mean by that? Description of the methodology. That means the procedure that you have done, how you are going to collect the data, how you are going to do, how many trials you are going to do. So that is why presenting sufficiently detailed information while avoiding unnecessary repetitive information. That means how you are going to do it, what are the ways of doing it. That is the description of the method for collecting. It's very clearly written, collecting or selecting the data. Okay, coming to the next, this one, I will now give you more clarity one by one. Now let's talk about this. Now, always understand that the aim is different than a research question. Aim is always in a sentence formation, okay, in a sentence. Whereas the research question should contain the dependent and the independent variable as well as it should be in a question form. So if I'm going to do a experiment on what is called as the transpiration, okay, this is transpiration. Can you see it? Okay. So,
Yes, yes, I can
yes.
see.
So now this transpiration, I'm going to do an experiment on transpiration. So it says that degree of effect of wind speed on different size plants and to test if larger leaves have higher transpiration rate than smaller leaves at higher wind speeds, where it is not that clear as to what am I going to. So here the independent variable degree, the degree can vary from one extent to the other extent. Different, I have not mentioned which plant, which, what is the size. So it is just the aim of what I am planning to do. Okay, now according to this, so one thing is clear, I am going to do on transpiration. One thing is that I am going to do with respect to the wind speed. Got that very clear? Now
Yeah.
what I am going to do is that now when these things is clear to me, I will have my background information with respect to what? Can you tell me, Smita? My background information should be with respect to what? The wind speed and the transpiration. So
Okay.
whatever I will find out the information from the net, I will do a research. Am I right? So
Hmm.
the research that I'm going to do, would I will be doing the research on the transpiration, correlating transpiration with the wind speed. And people who have done this experiment before, okay, I will put all the things, what is the information that I've collected in this background information with citing, referencing. Either it can be on the footnote or it can be in text citation. Okay,
Okay.
that is it. Now coming to the next one. Now if you see this one, I will show you. If you see this one, now can you see the background information? Okay,
Yeah.
now my background information, I should write this very clear. This is my background information. So can you see, I have written all the background information, whatever I have to do. Now this I was thinking of doing with the germination. Okay, so I related everything with related to the germination and I put it over here. So whatever, what is the detailed one? The prior, whatever knowledge I have got, I have put that. The biological theory, whatever is there. So I did a research, a thorough research. The species on which the different they have done it, like Pisum sativum. So the Pisum sativum is a pea plant. So what, how they have done it. Now
Did
there,
you do the primary research or any other research?
now primary research, yeah, this is the primary research that I have done it. So I have
Okay.
collected all the information, I have put it, I made a Word document, I put all the information over there. Then while writing the internal assessment, I finalized which I have to put it, which is relevant to my research question.
Okay.
So this is all the background information that I have done. Scientific sources. So I have mentioned here, see this odd ethnic 2011. So I got from there. So I have made in-text citations over here. Okay, multiple sources I have taken. So this is an example for you to see how the background information should be written. Background information should not be more than three-fourths of a page. Okay, because you should be also very clear that it should be coming within 3000 words. That should be always there. So the this should be very very relevant, that what is it that is coming in.
You will be able to share this here, ma'am,
Yes, I, yes, I can share it
Okay.
with you. Yes.
Thank you.
Now coming the next one. Okay. Now this is the research question. Can you see the research question that I have made? Okay.
Yes.
Now when I am making the research question, see, I said my wind speed I have specifically made. Are you getting it? Can you
Yes.
see? Okay, now you can understand the difference between the aim as well as my research question. Can you see?
Yes.
Directional, and I mentioned it very clearly. Now it is impacting the transpirational rate, how I'm going to measure it, and in which plant. Okay, and
Yes.
various Leaf surface area, they are small, medium, and large. Can you see this? So I am making it very, very clear. And how I am going to measure the rate of transpiration? By the rate of water uptake using a instrument called as photometer.
Mm-hmm.
So this is how my research question has to be there. So my independent variable is the wind speed. Okay. My independent, I will just highlight it also so that it is very clear. So my wind speed, this is my, sorry. So this is the wind speed is my independent variable, sorry, and this will be my the rate of this will be the dependent variable. Got that very clear?
Transpiration rate.
Yes, transpiration rate. So, how am I measuring the transpiration rate? With the help of rate of water uptake. That is how I am going to measure it. Okay,
Okay.
using a photometer. After this, I'm going to, once I have written, so what all I have written right now? I have written the aim, I have put my background information, and now I have written the RQ also. Now, the next thing that I am going to do is the hypothesis. Now, what is a hypothesis? The hypothesis is something which is my own thinking, the knowledge that I've got, that I will say, like for example, one at 1 meter per second, the transpiration rate will be low, but at 2.3 it will be high, and 3.2 again it will be low. That is what my hypothesis. It can be right. After I do the experiment, it may come the way I thought about it, or it may not come. It is okay. So hypothesis is prediction, what I predict. Okay, and then you have to give a justification. Now, how you're going to give the justification? By the theory. So you can correlate this by whatever you have put the information in the background information. Got that very clear? So you have to, you just cannot write that at 1 it will increase and at 2.5 it will decrease. No, you can write, but you have to provide an evidence to it. Clear? And this evidence has to be scientifically proven, or there has to be data from where you are saying that this is how it has happened.
Okay.
Okay. Now coming to the next one. Now, can you see this one? Now this
I have
is
one
how.
question here. Can I ask one
Yes,
question?
yes, please.
What is, why is it important to write actually a background information? How is it useful?
Because when you write the background info, first of all you have done a thorough research showing that how you have how have you come to the research question, what made you to come to this research question that you have chosen. Okay. Second thing, you have to do an analysis. After the data you get, you have to do an analysis. When you have to do an analysis, you are saying that this is going to happen at this temperature, or this is going to happen at this wind speed. How? Who said that this is going to happen? You have to substantiate it with a evidence. Okay, and that evidence, where have I put? In the background info. So I can relate, I can write over there, please refer to the background information, or it is there. You can write that author's name or the scientist's name who have done it.
Okay.
That is why the background information is very, very important. Now,
Okay.
can you see this one? IV is the independent variable, and dv is the dependent variable. Can you see it? Yeah.
Yes.
Okay. Now, this IV clearly states what is the experimental group. Now, in this case, the solutions apples were submerged in. So, what were the solutions the apples were immersed in? So, like for example, I took pineapple juice, I took lemon juice, I took salt, honey, and distilled water. So my independent variable is what? The lemon juice, the pineapple juice, the salt, the honey, and the distilled water will act as a control. Why I have to take a control? Because because control is something which, with respect to which I will understand whether it has increased or decreased. Now, I will give an example, very simple example. You know, pH. The neutral pH is 7. Okay. Let me say that the neutral pH 7 is the control. Okay. If anything goes less than 7, okay, that means it is acidic. If anything goes more than 7, it is basic. So 7 is acting as a control. Are you getting it? So now
Yes.
here in this case, when I'm taking different solutions, so what my distilled water will act as a control, on the basis of which I will understand or I will analyze whether my reading or whether the data that I've collected has gone up or gone down, and why it has gone up. Now, coming to the, and for every independent variable that you take, a justification has to be given why you have chosen it. Why did you choose pineapple juice? Why did you choose lemon juice? Why did you choose salt and honey? So, a justification has to be given. Now, coming to the dependent variable. In the dependent variable, what is the percentage you are doing here? Okay. Now, percentage change. Okay. For the whatever you take the dependent variable, justification for that also needs to be given. So, for anything that you do, a justification has to be given. Now, coming to the next one, I will talk about the controlled variable. Now, as I told you, we have to have one controlled variable, one independent variable and one dependent variable, and rest all has to be the controlled variable. Because if others we keep as control, then only we will understand is the independent variable, how much is the independent variable that we have taken is affecting the dependent variable. Otherwise, there will be a mix of everything, and we will not make a clear prediction, or we cannot get a clear analysis of the data, what we are going to do it. Now, always the control variable should be in a tabular form, which is preferred by the moderators. So the variable is written over here. If you see, all the variables have to be written here. The impact, what impact it will have on the this variable has got on the experiment, and how are we going to keep it constant, what are the way how we are going to keep it constant. uh, did you get it? Now,
Yes, yes.
concentration of this Ga3, this is gibberellic acid. So how I am going to keep it constant, this I'm going to write it here. And if I don't keep it constant, what would be the impact of it? Got it? So this
Okay.
is how you have to mention it very clearly. Now, the next part, that is the risk assessment. This is something which IB is very, very particularly looking into. What I told you about what is called as a safety ethical, I hope you're remembering. I
Yes,
told
yes.
you safety, ethical, and
And environmental.
yes, yeah, environmental. So that is how I'm going to. Now, NaCl, it is very, very irritating to our skin. So what is the hazard? Like skin and eye irritation. Okay, so what I have to do, I have to wear a goggles, the safety goggle, and the gloves I have to do. And also, I have to store this in a sealed volumetric flask with clear labels. So this is how I'm going to control it. Now, this solution, it can again cause a skin or eye, and again I'm going to keep. What are the ethical? If you see this one, now, this I have mentioned it very clearly how I am going to do it. So, eh, so I have written over here, regardless of the dangers within and with, this section needs to be present. This is very important. The risk has to be clearly identified and explained. And in some experiments, like if you say take secondary data, if the secondary data may not have the health risk, but And this should not be mentioned. If you're taking secondary data, then this should not be mentioned. But if you're doing an experiment, you're going to collect the primary data, then this is very, very important. And you see, I have written here very clearly regarding waste disposal. Waste disposal means the chemicals, how you're going to dispose it. Okay.
Okay.
Okay, now coming to the next one. Now, this is the materials required. I told you about the method, the procedure. Okay, now the procedure, when you're writing the method, you have to have all the material. What do you understand by the materials? Materials are what you are going to take, what all chemicals, what all things you're going to. So there you have to be very specific. Like I took 6 pairs of disposable rubber gloves. Okay, I
Mm-hmm.
took one vernier. See how many vernier lab pro pH sensor I took? I took one. How much did I take the garlic? Minced garlic, I took 1 tablespoon. Now, 1 tablespoon is what? It is 15 ml. I made that also very clearly. When I took 1 teaspoon, that means 1 teaspoon is 5 ml. 1 cup is 236.5 ml. So, can you see? This is very clear. This is what, again, the moderators are looking into.
Mm-hmm.
And you see, what is this? This is very important. Sorry. This is
How
very
much
important.
help can I expect from my teacher?
The teacher, actually, the IS, we do it in the second half of the DP1. We start by the February or March of the DP1. So you
Okay.
have got already 6 months already, because if you have
Mm-hmm.
starting the session in August, okay, and if you do it in the March, okay, now what happens? You have got almost 6 to 7 months. Now in that 6 to 7 months, the teachers, there is a thing called as, they have to do practicals. Okay,
Mm-hmm.
now in the practical activities, the teachers have to teach all these things. So that 6 to 7 months is needed for the student to get prepared for the IA. So the
Okay.
practicals, what they do, should be written. They conduct the practical, but when they write the practical report, they have to write in this manner.
And within how much time I have to finish if I start in March?
See, for if you start in the March, first of all, you have to formulate your research question. You have to find out the methods. So, in total, you can finish by July of collecting the data. I'm talking about collecting of the data.
Well, 10 hours you said includes collecting the data also.
Yes, yes.
Why do I
Yeah.
need July then, till July? 10
No,
hours can finish.
no, no, no. What I'm saying is that 10 hours is for doing the experiment. These all things are not included in the 10 hours.
Okay, okay, okay,
The
got it.
10 hours is solid practical work that you're doing.
Okay, got it.
The other things are not included in the 10 hours.
Okay.
So when when your teacher has to teach you in this manner, and when they teach you this manner, for you IA will be very, very easy to do it.
All these chemicals are provided by the school.
Yes, yes, all this,
Okay,
it is all present in the school. Okay,
okay.
now this is the materials and the apparatus. Now if you come, how am I going to prepare the NaCl solution? This is the preparation part. This is what I needed, all these things I needed, like kitchen knife, cutting board, everything I needed. Now how I am going to make it? So I need how much grams of NaCl. I've made it very clearly. Okay, how much, what I need, an electronic balance to measure it, how the beaker I need, how much distilled water. So this is where I'm going to prepare it. So my this is very clear how I am going to do all the things. Okay, now I was talking about this plus minus 2.5 ml. Everywhere there's plus minus. What is plus minus? This is the uncertainty. Every equipment will have an uncertainty. What is uncertainty? Like, for example, I have predicted a mark for you like 20 marks out of 24. So you may get 18, you may get 21, plus minus. Okay, that is uncertainty. So this uncertainty is least count upon 2 of any. If you see any apparatus, okay, every apparatus has got, or equipment has got, a least count. The least count divided by 2 is the uncertainty. That is how you calculate it. That is how you write it down, or you calculate it.
Is
This is
is uncertainty essential in this IA?
Yes, yes, yes. Yeah, IB has made it very clear that uncertainty has to be there all the time,
Okay.
even biology and ESS. Earlier,
Okay.
it was only for physics and chemistry, but now they have made it mandatory. So, least count divided by 2 is the uncertain. That is how you count it. Okay. Now, coming
Okay.
to the next one. Okay, so this is all you have done about the research design. Okay, so everything you have seen, I will go once again to you. Okay, I think I missed one. I'm so sorry. Methodology. So after, so you saw the risk management you saw, materials required you saw. Now we are coming to the procedure. Okay, now can you see how I have written, how I am going to, last slide showed how I'm going to make the slides. Now I have put everything properly, how I am going to do the procedure. Now, when you're writing procedure, Smita, you should understand that the procedure should be written in such a way, step by step, that when the moderator reads it, he can understand, okay, this is how it has to be done, this is how it has to be done, and I can do it exactly, and I will get the result. So the moderator should, as soon as he or she reads it, it should run in his mind that, okay, after this step, this he or she, the candidate has done. After this candidate has done, and this is how the candidate got the data. So it has to be systematic wise. See, that's why I wrote very clearly. It should be specific with apparatus, all the apparatus, how you are going to do. Like you see, using the measuring cup, measure out half details of go together and one small this. So I'm making it very, very clear over here. Now, the rule of thumb is very clear. If it's not written, it is not done. Need to be very, very explicit. Now, if you don't do it, it's not that the moderators will not will not read or anything of that sort. They will deduct your marks of it, because it is total 6 marks, and there are 3 aspects. Okay, 2 marks for each aspect.
Okay.
So 6, 2 into 3 is 6, so they will deduct the marks. Okay, now a good example, as I've told you, pour 40 ml water into a 50 ml graduated cylinder plus minus 0. This is how you have to write it. Now if you write like this, pour water into a graduated cylinder, what does the examiner understand, or the moderator understand? So they will deduct the marks. Okay, and you should include the diagram and your experimental setup for the clarity. If you're doing this, so relevant to what you've written, the picture should be there. Your face should never be shown. That is another IB wants it.
Mm-hmm.
Your face should never be shown on the in your record, in the record, when you write the record. Okay, so like this. Now when I'm saying that I have to mix it, I'm mixing it over here. Okay, so whatever I've written relevant to that, I've put the pictures. Okay, if I have adopted a procedure from some research paper, I have to refer it, that this is from where I have taken it. If not, then I write it down clearly. Okay, so
Okay.
with this we end with the research design. Okay, now
Okay.
coming to the next one. Now the second criteria, that is criterion B, that is data analysis. As I told you, this also has got 3 aspects. Okay, each aspect will be of 2 marks, and that is why it is of 6 marks in total. Okay. Now I am going to record the data because now I did the experiment. Okay.
Okay.
And I will record it. After I record it, okay, I will do the processing of the data. What do you mean by processing of the data? I am going to put it in some formula. The raw
Mm-hmm.
data, if I record, the data that I'm recording is called as the raw data. I cannot use the raw data like that. I have to process it by putting into a t-test or a chi-square test. Somewhere I have to use some method. Statistical, I have to use the method. And the table should be clear. The communication should be very, very, I will show you again the sample, you will understand it very clearly. The table, see, I said graphs, tables, use of units, decimal places, significant figures should be very, very clearly mentioned. Okay. And again, all the processing data should contain the uncertainties. If you don't put this uncertainty, your marks will be lost. So most of the students lose marks on this uncertainties. Okay. The inaccuracies or inconsistencies has to be, they are told very, very clearly. Now if you see what is the difference between both of it in this, now here there is major omissions, here there is significant omissions. Okay. And this allowed the possibility of drawing a conclusion. Okay. That means what? There is a significant omission which may allow not to come to the conclusion that is being desired. It may give you a wrong conclusion. Okay, now I'll get when I explain this with using this example, you will understand in a much better way. Okay, now let's come to this slide. Can you see this one?
I can see.
Yeah.
Thank you.
Now the raw data. Can you see raw data? Now
Yes. Yes.
raw data. Now this is how the raw data has to be. So the table has to be first, I mean, labeled very clearly. What am I going to do? So I have taken the initial and the final pH using what? The pH meter. I have taken the pH meter. Okay. From here, I have taken the pH meter. And then there is a difference between them. Okay. So when I'm doing it, I have to clearly tell what exactly is it. And then here, if you see, my table is so clear. I have written over here the trials. I have written here the concentrations. I have written here the initial pH. So every trial, this would be the initial will be one single and the final and the difference. This delta shows the difference. Okay. And can you see now, do you know how the 0.02 came? Because I'm going to minus final minus pH, that is the difference, and this is 0.01 and this is 0.01, that is why it is 0.02. Got it?
Okay.
I mean, you can ask questions, can you, if you don't understand. See, how do we calculate difference? We calculate difference from the final minus the initial, correct?
Yes.
Now, so we have, so 6.41 minus 6.68. So that is why my answer is coming in negative, correct?
Okay.
Because my final is less than the initial, correct?
Yeah. Yeah.
That is why it is coming in minus. And why this is 0.02? Because this uncertainty of the final pH and this uncertainty of the initial pH, I added and brought it like this. Got it?
Okay. Got
So
it.
this is trial 1. Now this is trial 2. Now trial 3, trial 4. Like that it goes on. Got that very clear?
Yes.
Okay. Now, after this has been done, what did I do? I have taken the average of it. Okay. This is again, I have done it for the average. So, like this, I have to keep it very, very clearly. Now, why I have highlighted this? Can you tell me? if you can just look into it and tell me why I have highlighted it.
You mean to say the orange color?
Yes, this one. Yeah.
The number is not clear on top of that.
Okay, this is 82
Okay.
and this is 46.
Okay.
Okay.
Yeah,
Why? Because
yeah, I think it's everything is in one band and it seems to be different completely.
yes, very good,
Like
Smita.
an
Because
outlier.
yeah, because it is an anomaly. It is
Okay.
not in line with it. Very
Hmm.
good. Okay, so that is out. So the raw data should be separated from the processed data. So this is the raw data and this is the processed data. Got that?
Yeah.
Okay. So units and uncertainties are listed. All data must be presented to the same accuracy. Now, how? I'm telling you. Now, how many after decimal there are two figures? Am I right? Okay.
Yeah.
So here also it is two. If the answer was, see, can you see here 0.9? So you have to put a 0.
Mm-hmm.
This is called a significant figure.
Mm-hmm.
If your answer, I will repeat once again, if your answer here, like it is coming 9.9, okay, so you
Mm-hmm.
have to put 9.90 because here after point there is two
Okay, two
digits.
decimals. Got it.
Okay, if it was 9, so Smita, what I should write? If it is 9, 9 point.
9.00.
Very good, Smita. That's
Mm.
good. Very good. Understood? So
Yeah.
this is what I was telling, to be to same accuracy. Okay? This you have to do it. Clear?
Okay. Clear.
Okay. Now we go to the next slide. Now, I'm going to show you how I have done the processing of the data. So what I got over there, the concentration I've written over here, I've written the change in the dissolved oxygen. I mean, how? The change is what? The final minus the initial. Now, percentage. You know how to calculate percentage. This change upon the initial into 100 would be the percentage. So this
Okay.
is what I'm doing is what I'm processing the data. So I have to show one sample calculation. What do you mean by sample calculation? Like how I got this. Okay, if one I show, then they know that this you have done in the same manner, and this is very important.
Okay.
Okay, so sample calculation has to be shown. Sample calculation has to be shown. Okay,
Okay.
yeah. So now coming again. So here I've written the concentration. So I hope you understood change. Change means final minus the initial. Okay, final
Okay.
minus the initial. Okay, okay, and
Yes.
what is the percentage? So this one, whatever I've got, okay, the change, okay, this is called as the change, am I right?
Hmm.
Change upon the initial, okay,
Times 100.
into 100. Very good, Smita. Okay, so that is a percentage. Okay, and the change pH is what I have got in the raw data I calculated over there in my previous one, that I have put it over here, and I will show how I have done it. Okay, so here is again the mean. I have calculated the mean. So this is the standard deviation. Now standard deviation, what I will tell Smita is that you can use what is called as the GDC. You know what is GDC,
The
Smita?
calculator.
So in the standard deviation, you can use the GDC, and what you can do is that you can take a picture shot and you can put it. Okay,
Oh,
that is
okay.
how the standard deviation and coefficient of variation can be taken, also been done with the GDC.
Okay.
Okay, okay.
Okay.
Now come. This is the process data. Now coming to the next one, that is the graph. Now, one main thing that you should understand is that the graph that you make should be of the processed data, not of the raw data. Make it very clear. I
Okay.
repeat, the graph that you are going to make, this one should be of the processed data, the one that you have calculated last in this one. Okay, it should be of the process data, the one that you have done, okay? Not of the raw data. If you make of the raw data, it will be yours. The examiner will give, will not, he will put a cross over here, not accepted.
Okay.
Okay, the graph should contain the caption. Can you see? If you don't put this caption, if you don't put it like this one, what is it? The examiner doesn't understand, and there will be reduction in the marks. So these are the where the graph should contain a detailed caption. Axis should be labeled. Most of the children don't label this axis. Okay, and also this should be there, the unit should be there. Now here there is no unit because it is the type of the fruit peel. If there is capsicum mass, so the what is, how is it been measured? The milligram, or this has to be, unit has to be there. Okay, the scaling has to be appropriate. That means the interval should be same, like 30, 35, 40. You cannot write 30, 35, then 45. So the interval should be the same. The interval should be the same. Got that very clear? Now, if you see here 10, then it is 20. You cannot 30. If you put 10, 30, then it should be here 50. Got it?
Okay.
Whatever you put, it should be the same interval. Okay?
Yeah.
If you're taking a screenshot, it should not be blurred, but it is better that you make the graph using an Excel sheet. How you're going to make the graph? Using an Excel sheet. Got that very clear? Excel
Yeah.
sheet, you will use it. You will not take a snapshot. That is not right. Okay?
Okay.
Okay. Now, coming to the next one, the analysis. Okay, now you're going to do the analysis. Now the data you have got, raw data you got, you process the data, you put the processed data in the form of a graph. Now you're going to analyze it. Okay, now when you're doing this analysis, you are going, whatever data you are going to get, you're going to analyze by writing the value along with it. Like as you see, as seen in graph 2 and table 6, you have to mention the lemon peel has the greatest biopsin efficiency at 73.4. Are you getting what I'm saying? So
Yes.
the graph has to be mentioned, the table, from where are you writing that this has increased, and to what extent? Okay, anything do you write, like the banana peel biopsin efficiency is 39.2, lower than. So if you write it is lower than the orange peel, how do the examiner under how much is it lower to what? So you have to specify. Clear? So
Yes.
this is how you're going to do the analysis, and when you're writing it, you have to give, this is supported. Can you see? I'm supporting it by this test, see appendix 6, like this. Are you getting what I'm
Okay.
saying?
Got it. Got it.
Okay, so like this. So you have to use a statistic t-test or chi-square test, as I mentioned earlier, and this has to be done.
Okay.
Okay. Now coming to the third criterion, that is the conclusion. Okay. Now in the conclusion, again, we have got the aspect. Okay. There are two aspects of it. Okay. The conclusion is justified. So you have done the analysis. Now you're coming to the conclusion. This is what you have with respect to what your research question, okay, and the analysis that you present. You have to give a conclusion which has to be justified. Okay, and this justification should be relevant, and it should be with respect to the scientific context. Now, where did I write the scientific context? In the background information. Okay, refers to the information. Can you see here?
Yes.
Which I
Yes.
have done it. So that is why the background information is very, very important.
Madam, the meeting will end in 5 minutes, is what is showing here.
Okay, fine. The other thing is that now we come to the next one, how you're going to write the conclusion. Can you see the conclusion? I have written it very clearly, how on the basis of the thing that we have done in on the analysis and everything. So I restate the RQ, I readdress the hypothesis, I discuss the strength of the conclusion, and I readdress the context. Everything, whatever I have done, I'm concluding, it's like a synopsis. Okay, I've done it in this manner. Okay.
Okay.
And then the last one is the evaluation, okay, which also contains two aspects. Now, in the evaluation, as the name suggests, you have to write the strengths of your this one, the weaknesses you have to write, and the limitations you had. Okay, and how, and the improvements, how you are going to improve. If I have to do this experiment again, what all things I should take into consideration and how I can improve it. Okay, now
Yes.
here again, can you see this? I have put it very clearly. I'm going to share this with you so you can see the strengths I have written, all my strengths I have written. So these are all my strengths when I did my experiment. So the intention was to validate the relationship. So this was there. So trials was there. Potential applicability was there. So all these are the strengths I have written. Now, the next slide will show the weaknesses and how to improve it. Always remember that the moderator wants it to be again in a tabular form. So, whatever weaknesses you have written or limitations you have written here, how you are going to improve if I have to do the experiment again. So, this one, this, whatever you're writing, should this, the improvement should be in accordance with this. You cannot write anything else. Got that very clear?
Yes,
So
yes.
this, both sides should be relevant. Okay, and how you can extend this experiment if I have to do a further research on it. Okay,
Yeah.
these are the things, and this is the work cited. I have done it. Okay, I have put some examples of the what is called as the RQ. Okay, what that a candidate or you can do, you can look into it. Okay, and
Okay.
also some common mistakes that happens. Okay,
Okay.
that also I mentioned it for your reference. Okay, so there are common mistakes that is there and how it can be rectified. Okay, and there are the control variable, all those I have the common mistakes that are there, and that is all about it.
How is this IA different from external essay?
Yeah, this is a very good question that you have asked. Okay, let me stop sharing the screen with you.
Okay.
Okay, so then okay, yeah. So the what is it is that